Better. Great. Great. I don't have pandas in my talk either, but I have this image that I like to look at, which is a human cerebellum scanned at 150 micron. So this is a postmortem scan I took at UCL in a 9.4 Tesla scanner. And I like to look at it just because it reminds me how fantastically folded this structure is. This is a structure that contains more than half the neurons in the brain. And unfortunately, with all the available databases that we have, we don't really have that many data sets that would be suitable. We know that the cerebellum is very functionally heterogeneous, and it subserves a large number of not only motor functions, but also cognitive functions. It's functionally heterogeneous, probably as diverse as the neocortex. And as I said, current databases have a number of problems. The point database has a problem that we often, the cerebellum is often not fully covered, in some subjects covered, but not in all. And it's not reported very easily where we're actually missing observations. The different subjects, different baselines that make inferences very hard. And even more so than with the neocortex, there are often very different atlas methods and inaccurate localization. We'll come to this point later. So we thought in our lab that it was really time to collect a larger and a more extensive data set on a comprehensive task mapping of the human cerebellum. So the problem with functional imaging of the human cerebellum is that at current resolutions, even if you go down to sub-millimeter functional resolution, it's very hard to resolve activity coming from these individual folia. So what we've done over the last few years, and this is all open source pipelines that are available, we have worked very hard on doing a very good volumetric normalization of the human cerebellum. We have, for example, included in this package a flat map that is just a hull of the human cerebellum that serves to visualize these visualized activity patterns in the human cerebellum, and we've also provided a probabilistic atlas that gives you the opportunity to label your data on the normal kind of standard anatomical annotation in the cerebellum that comes from Lasso that labels the cerebellum in lobulus running from 1 here in the interior cerebellum over 7, cruise 1 and cruise 2, down to lobulus 10. And this is how we often think about localization in the cerebellum. Now, in the data set, we really decided to throw the kitchen sink of cognitive neuroscience at the cerebellum. So we have, right now, we have 17 subjects that we scanned in 4 sessions, so about 6 hours of scanning per subject. We have experiment 1 with 29 task conditions, experiment 2 with 32 task conditions, and an overlap of 14 tasks that were repeated across the experiment to really have a good measure of reliability here. People did all these tasks within a single run, so that we really have a common baseline for these. We have a 5-second instruction period and a 30-second task period, where there are often multiple task conditions within those 30 seconds. So this just in the cerebellum now gives you just an impression on the richness of this data set. What I've done here is I've picked all the shared tasks and gave you the average activity map from the first experiment and from the second experiment. You can very nicely see that we can very rapidly elicit these activity patterns. We get, for example, the motor network in the cerebellum that consists of lobulus 5 and 6 up here in the anterior cerebellum and lobulus 8 in the inferior cerebellum. But you also can see all these cognitive tasks here that they elicit different highly reproducible activity patterns in the human cerebellum. So we can take these tasks and we can now try to figure out what are the features that really underlie the cerebellum. So what are the cognitive features that the cerebellum cares about, also especially compared to the neocortex? This is a multidimensional scaling plot where we look the similarity of the activity patterns. So each activity pattern for a task is a dot in the space, and the distances between these dots are a representation of how dissimilar those activity patterns are. So we have, for example, here the kind of working memory tasks that activate these like lateral cruise networks. We have tasks that involve visual attention and eye movements. We have tasks, theory of mind is a very thankful task to drive cerebral activities. It's kind of this medial network. We get replicate old studies on verb generation with kind of a right hemispheric network in the cerebellum. We have action observation is very nice in driving these perimotor networks. And that's very interesting compared to the neocortex, one class of tasks that don't drive cerebral activity, maybe not very surprisingly, are looking at complex visual stimuli. So that's very good for inferior temporal cortex, very good for visual cortex, but the cerebellum really couldn't care less about this, actually cares more about it sitting there and just resting. So this is the cerebellar equivalent of the default mode network right here. So to summarize this, we can now try to extract some factors. And so what we've done here is a simple ICA on the evoked activity patterns, and we can kind of isolate these different components. We can now go in and label these and just look at how these tasks now load on these different components. Here are two examples. So I picked these clear left and right motor networks here, just an example. What you see here with a plot, all the tasks in the space of component 1, and which is this network, and component 2, which is this component here, you see that they fall very nicely. We saw some of the tasks we pointed to a left-hand response, some of the tasks to a right-hand response, and the tasks that activate both of these components have a bimanual response. And here are the tasks that actually don't involve manual responses. So we can go through now and kind of look at the functional profile of each of these components. And you see the motor networks. We have a clear ocular motor or attention network in the middle. We have an action observation network, verbal fluency, right hemispheric. And we have a number of different components that are functionally related that all kind of relate to more higher order executive function and the lateral components of cruise 1 and cruise 2. And we have these two interesting, more medial cruise 1 and cruise 2 networks that are really caring about thinking about other people and biological motion. So there's a little bit hemispheric specialization here for looking at motion stimuli or processing verbal stimuli in the context of social cognition. So the goal of this is to make this data set available, but also the goal is to see whether and how, especially, we can predict cerebellar activity patterns for novel tasks. So this is why we have two different experiments with two different task sets. And we can do that either from the characteristics of these tasks or actually just from the activity of the cerebral cortex. And if you're interested in discussing that with us, we'd love to hear your feedback. We'll be available at these two posters tomorrow. Before I end, I just want to point out one thing that we definitely learned from this data set already, and this is actually quite an important point to make, I think. So as I mentioned before, traditionally in the imaging literature, when you look at results tables in NeuroImage or something, if people report cerebellar activity, they say, oh, I found activity in lobulus cruise 1 or in 6, if you're lucky. Otherwise, they just say, well, I found cerebellar activity, which is just as good as saying, oh, I found cortical activity to me. Now, the problem with this is that when you look at these functional components, they don't really fall clearly along these lobular boundaries. So these macroanatomic boundaries might not be functional subdivisions of the cerebellum. Indeed, they seem to be quite unrelated here. And to test this formally, we just calculate the cross information of these two maps and compare that to calculating cross information between this map and just any random continuous tessellation of the cerebellum. And it turns out that when you just look at this metric, which is just a cross information metric here, the distribution of random tessellations does actually as good or better than the one that has the lobulus. So the strong conclusion here is that lobular boundaries, as apparent they are and as invariant they are in the anatomy, and therefore those are the only things that we can actually have in the cerebellum to really say where we are, they don't constitute functional boundaries. So really, we need to stop using lobular denotation. We need to use our flat map or other atlas methods to really tell us where exactly on the cerebellum we are. And new strategies for meta-analysis and integrating these results are needed. I just want to mention my lab, thank my lab, and especially Mev Kang, who was an amazing graduate student who collected this data set. I want to thank you for your attention.
Thank you very much. We have time for one question. Is there over there?
Yeah, this is not a question, it's a comment, but I really want to congratulate you on testing a null model on parcellations. I think if many people did that, their results would fall. So, I mean, thank you so much. We need more of this.
Thanks. Okay. Well, you can thank my graduate student who did that.
Can I ask a question? Thank you for the nice talk first. Then you recommend using the surface representation for the cerebellum, but you know, on new subjects, it's maybe very hard to get the surface right, just because it's so tight. So you might have lots of projection issues when you project the data.
Yeah. The surface that I showed and that we've released in our open source toolbox is not a real surface. It's just a hull. And that's the same after volume normalization. It's a totally cheat surface. It's nothing like the neocortex. This is a real cerebellar surface. So as Marty Serino made this from our scan, you need a scan of about 150 micron resolution to do that. And it took about half a year to flatten that thing. And it's 1.5 meters long and 15 centimeters wide. So you can imagine, like in the journals, you kind of need the centerfold folder. That's kind of my dream, if I'm there. But unfortunately, functional scanning is, even if we go down to 400 micron resolution, 200 micron resolution, you still can't reliably resolve individual folia, and that will always be a problem with using flat representations for cerebellar data. Otherwise, I would love to do it. Yeah.